Welcome. This is Anna here, and we're going to talk about the neurophysiology. So we're moving on to this chapter 12. I think it's called chapter 12. And we're going to go over the nerve cells in this part 1 first and some of your histology, and then we'll start moving on. All right. So the nervous system is a conglomeration of different cells plus interstitial fluids, and also, you throw in the interactions with connective tissues and then the other things that they are taking things to. So let's look at this in terms of kind of a flow chart of functions. So sensory section, so let me back up. One of the things the nervous system is going to do is collect information. So it's going to collect stimulus, and this stimulus could be from inside the body, okay, or it can be from the outside environment, but it's going to collect that information. And then that information is going to go to an integration center. And this is where you're going to process the information, you're going to interpret the information, and this is going to be done primarily in your spinal cord and brain, okay? Once you process information, you want to do something with it. So the next thing that's going to happen is motor output, okay? And motor output, I actually don't want to do those little arrows. Motor output, you're going to send out commands, information, commands, etc. And it's usually going to go to some type of effector organ. Okay. Examples of effector organs would be muscle cells. Okay. It could be glandular epithelial cells. Okay. It could be other nerve cells. Okay. It can be a lot of different things. Okay. Now, this looks like it's a very linear thing, but remember, motor output is then going to be picked up by sensory receptors as well. So saying what happened outside your body, where did you move in your environment, what happened inside your body. Okay, so it's really more of a looping mechanism rather than a strictly linear mechanism. All right, next slide. All right, let's make a messy map, okay? We need to organize the structure of the nervous system, okay? And the nervous system in and of itself, when I think of it, I then divide it out into what is called the central nervous system. So that's central, why am I doing it this way? Okay, we'll just write it central nervous system, and the abbreviation for that, which I'm going to use from now on because my handwriting is terrible, is central nervous system. And then you've got your peripheral nervous system, and that we're going to abbreviate as PNS, okay? Now there's going to be some overlap in how these things work, okay, and how we define and name things, but this should get you started, okay? The central nervous system, we're going to then think of in terms of brain and spinal cord, okay? Those are the two major organs of the central nervous system. But both of these are going to have bundles of neurons that we're going to call tracts, okay? And a tract is a bundle of axons in the central nervous system, okay? You're also going to have, feeding into those tracts, the cells, okay? And the main cells are neurons, but then there are several glial cells that we're going to talk about as we go further on, okay? But that's basically how it's organized. Now, the peripheral nervous system, we can organize in a couple of different ways, okay? I'm going to first, and you could do it in another way, but I'm going to go ahead and divide it into sensory and motor. Okay, now it's anatomy. We need multiple names. So we can divide that into afferent, or also call that afferent, and this is efferent. I focus on the letter E, so E for exit. Okay, that helps me keep them organized. Okay, now what's interesting is that we can also think about it in terms of somatic, which is body or voluntary, and we can think about it as autonomic, which is involuntary. And from now on, I'm going to write autonomic as A and S. Now, with these two groupings, each one of them has motor, and each one of them has sensory. Okay? So that is another way of thinking about it. Okay? Now, part of the central nervous system, the peripheral nervous system, is that it is controlled by nerves. That's its dominant feature. Now over here, I wrote down tracts. Tracts and nerves are the same thing, but location matters. All right? If you're talking about a tract, it's in the brain or spinal cord. If you're talking about the nerve, it's everywhere else, the peripheral nervous system. And again, the definition, bundle of axons in the peripheral nervous system. Now, I have two types of nerves. We talk about spinal nerves, and that is going to give rise to the bulk of the named nerves that we're going to talk about eventually. Okay? Now, spinal nerves originate off of the spinal cord, so their origin is on the spinal cord. Okay? Cranial nerves, their origin is off of the brain, and we have 12 of them, and we will eventually talk about them. Okay? So you've got central nervous system contrasted with peripheral nervous system. Okay? You have nerves contrasted with tracts. In the peripheral nervous system, we can talk about the sensory division or the motor division. Okay? We can also talk about it in terms of somatic nerves versus autonomic nerves. And I have somatic, sensory, somatic motor. Okay? And I have autonomic sensory and autonomic motor. Okay? And all of these can either be bundled into a spinal nerve or into a cranial nerve. Okay? Over here with our tracts, you will also have sensory divisions and motor divisions. Okay? All right, let's move on to the next slide. All right, what I want to talk about now are the cells. Okay? We've got the big, fancy, major cell that basically is going to be responsible for transmitting signals all over the place. Okay? It goes the distance, it gets things all over the place. But over here, we have a different type of nerve cell called neuroglia. Oops, misspelling that. Okay? Actually, so cells of the central nervous system and the peripheral nervous system. Now, your book does something that I think is irresponsible in that they often call neurons nerve cells. I find that misleading and I find it confusing. Be careful when you see that. Make sure you actually know what is being discussed and that you don't confuse things, okay? So you got the neurons, all right? The neuroglia, we got different kinds, and they can also be called glial cells, all right? The neuroglia, these are the support cells of the nervous system, all right? They support the neurons, they support other parts of the nervous system, all of that. There is an interdependence between the various glial cells and the neurons. You cannot have one or the other. The neuron is not the boss, all right? It is just one part of the nervous system. And in fact, there are some places where you could make an argument that the glial cells are functionally more dominant, all right? Neurons are highways for signals. Glial cells are the people who maintain the roads and the stop signs and the stoplights, and they repair the potholes. They're the ones that actually keep the roads functional, okay? Now, I can divide the glial cells into two categories, those found in the central nervous system and those found in the peripheral nervous system, all right? In the peripheral nervous system, we just have the two Schwann cells and satellite cells, okay? In the central nervous system, I have astrocytes, microglia or microglia, you can say it either way. I have ependymal cells, and I have oligodendrocytes. Okay, and we're going to start talking on the next couple of slides about these. All right, so first off, let's just go over our neuron anatomy, okay? So the neuron, which your book is also calling a nerve cell, but you got to be really careful because a nerve is not a neuron, all right? A nerve is a bundle. Let's just put that over here. A nerve is a bundle of axons in the peripheral nervous system. The problem with calling these nerve cells is that people then start to think that the nerves hold an entire neuron, and they do not, okay? Neurons, they are long-lived, generally amitotic, meaning they don't go through mitosis. We're now, 10 years ago when I taught this class, we were very firm. You die with the cells you were born with, and you never make new ones. We are starting to see in research with mice, and now they're looking at it in humans, that it is possible to develop and generate new neuron cells, although it's very difficult and very slow, okay? Another key component of neurons is that they have a high metabolic rate. We need a lot of energy to run our brains and nervous system. In fact, they are the most energy hungry part of our body, okay? Now, we got a bunch of different parts, all right? And if you look in your PowerPoint, I have a definition slide, okay, in the PowerPoint. And I'm going to talk about these on the picture, so I'm not going to go through and rewrite them out. But there are two definition slides that you need to remember that are there, okay? And I'm going to now just go on to the picture and talk about that picture. All right, let's look at this neuron, all right? We have the cell body right here, which can also be called cyton. I have right here an axon, okay, that runs the length. This area right here is called the axon hillock. And then down here, these things are the axonal terminals, all right? Now, if you have a myelinated axon, you have these things that look like white pearls strung on it, or white beads. These are Schwann cells, and they are responsible for creating the myelin sheath in the peripheral nervous system, All right, we'll talk more about that in a little bit. In between each of these, we have what is called the node of Ranvier. Okay. All right. So this is the axon that goes with this. All right. One more thing that we have are these structures here. These are called dendrites. Dendrites receive, axons send. These secrete neurotransmitters. Now, when people talk about the processes of a neuron, they are talking about these two things, dendrites and axons. So neural processes, so PR process, neuron. All right, those are the processes of the neuron, okay? And that just means a projection, all right? So what are some features that are important? Actually, let's do a drawing. Let me move over to another. No, let's not. Let's not. Y'all hate my drawings. Let's look at this picture right over here. Okay? So we're going to look at this next. And what I want you to notice is right here in the blue, that is the neuron cell body. And then I've got these long pink lines. Those are the processes, okay? Most of these are axonal or dendritic. Actually, all of them are axonal or dendritic. When you're looking at a microscope slide, it's very difficult to figure out which one is the axon. I happen to be able to see the axon hillock in this particular picture and see that it's thicker here and then it goes on. So I can actually say that that is the axon, but usually I can't tell the difference between an axon and a dendrite on a microscope slide, and I wouldn't expect you to either. Now, I want you to notice these purple dots right here, or pink, dark pink dots. Those are various cells, all right? Probably mostly in this case astrocytes, but there might be microglia in there as well, but not satellite cells because it's the wrong tissue type. And then in the background, I want you to remember when we were looking at the connective tissues and how there was a background that everything was floating in. That is the interstitial fluid here. Of your brain tissue, okay? And so you'll have that in there, okay? Okay, so you got your note slides. I'm going to go ahead and move on to the next slide. We're going to continue to talk about functions as we get down into functional zones and stuff. Okay, so I like this slide because it's showing you the different glial cells that are present plus neurons, okay? What are they doing here? Now, this is all central nervous system, so you can think of this as brain or spinal cord, all right? The reason I know that this is brain or spinal cord is because I have ependymal cells, which are secreting cerebral spinal fluid. I have oligodendrocytes, which are creating the myelin sheath, all right, that goes around the axons. I particularly like this slide because you see the neuron here and you see the neuron here. And then you can see the relationship that the astrocyte has between the neuron and the blood capillary, helping to regulate the environment, clean up neurotransmitters, and so forth. I can see here an axon, and here's an axon, and here's an axon. And this is an oligodendrocyte, and it's got little arms that come up and myelinate a certain segment of the axon. So here's another one, and it's myelinating here, here, and then here, and here, okay? And then you can see how they've cut open the myelin sheath right there. Right here, I've got a cute little microglia cell floating around. I like the ependymal cells right here. Do not confuse these with epithelial cells. They are not epithelial cells at all. Zero relationship with epithelial cells, but they do kind of look like epithelial cells. So they're kind of boxy. They've got these little arms that kind of extend out and increase surface area. And then it's got cilia right here that, oh, these don't increase surface area. I'm sorry, I misspoke. So you got cilia right here that's going to wave and move the cerebrospinal fluid. These little fibers right here are kind of anchoring it into the interstitial fluid, interstitial stroma of the brain or spinal cord. All right, let's look at another slide. All right, what I've done on the next couple of cells is given you a picture and then all the information you need about it, okay? So right here is an astrocyte. It's kind of a star-shaped cell. It'll have little arms that wrap up by neurons and then little arms that wrap around capillaries. Now, so it's anchoring these two structures together, but more importantly, what it's doing is regulating the environment here, okay, and here. It mops up extra stuff. It helps with the transport of nutrients and waste and all kinds of really important things. So it's super important, and that's what it does. All right, right here, you're looking at the microglia, all right? They also are monitoring the local conditions. They can become phagocytic when they need to. So the brain is not supposed to have white blood cells in it, okay? We're finding out now that that was a bit rigid in our thinking. We do have white blood cells that get in there when it's not pathological, but it's a very low number of white blood cells. So the primary phagocytic cell that works to clean up debris is the microglia, okay? All right, next slide. All right, here's some cute little astrocytes. You can see they've got this little star shape to them, which is why they're called astrocyte. All right, next slide. And you know that I love to show microscope slides. So here you can see another astrocyte with all the little starry projections coming out of it. Okay, next slide. All right, let's look in now at our ependymal cells. So what you'll see is this box shape to most of it. Right there, you will see cilia, and their job is to move cerebrospinal fluid. So they create a current that circulates the cerebrospinal fluid. And then it's got these little doohickeys that extend into the interstitium of the brain and anchor those cells into that area, okay? And the ependymal cells are going to be secreting the cerebrospinal fluid. So cerebrospinal fluid's abbreviation is CSF, okay? They do some other things, but that's like the basic thing that I want you to focus on. So here you can see kind of the shape of the cell. All right, there's the cute little cilia. All right, you can't really see the little anchoring things, but they're there. Okay. Now over here, we're looking at an oligodendrocyte. And what you need to memorize is that they create the myelin sheath, specifically in the cerebral nervous system. All right, next slide. All right, let's look at some histology. I've got a big old lumen right here. So this is the lumen filled, with cerebrospinal fluid. These are the ependymal cells, but I guess what you can really see is the cilia, and then you can see that they're staining kind of a darker purple up in here. Okay, that's it. Next slide. All right, let's look at the peripheral nervous system's glial cells next. We've only got the two. We've got satellite cells and Schwann cells. Well, let's look at the Schwann cells first. So first we're looking at this neuron. So this part, remember, is the axon. That is the cell body. All right, and each one of these little white beads is a Schwann cell. All right, what you memorize about that, they make the myelin sheath in the peripheral nervous system. These Schwann cells only go along the axons, and basically they insulate the axon to prevent the loss of the electrical signal. And we're going to talk more about this in the next little show where we talk about the myelin sheath. We'll eventually get to talking about saltatory conduction a couple parts later on. Okay. Now, if this was right here, the cell body of the neuron, I've got another cell type. I have these cells. These cells wrap around the cell body, and they are called the satellite cells. Their role, their job is not well understood, but the latest stuff that I've read this last year is it seems, whoops, what word did I just write there? Seems to be similar in function to astrocytes, okay? But you only find them in ganglia surrounding these sensory neurons, okay? All right, that was the last slide for part 1. So I'm going to stop the recording and you can consider looking in at part 2.